Okay, so today we're going to be doing the bleach lab, and basically what we're going to need is we're going to try to test what percent of bleach is effective against killing E. coli. So I've got my E. coli broth here. I've got a 10% bleach solution. Basically, I diluted bleach, or a 1% bleach solution. I diluted bleach, put 99 milliliters of water and 1 milliliter of bleach to get 1% of bleach. And then we're going to do our dilution. We basically want to input and dilute the bleach further to see which concentration of bleach is going to be most effective. So what we're going to do is we're going to have a 0.1% concentration, a 0.01%, a 0.001%, 0.0001%, and then just our control, which is our water. So what we want to do is I've got 9 milliliters of water in each one of these test tubes. So what we want to do is by taking the 1%, if we take 1 milliliter out of here and put it into this first test tube, it'll actually dilute it to that 0.1%. So all I'm going to do is on the pipette here, right at the top, it actually says 1 milliliter. So I'm going to take 1 milliliter of that bleach solution, and I'm going to input it into that first pipette, or into that first tube. Once I'm done with that, I'm going to put that pipette away. I'm going to take this test tube, and then I'm going to shake it. Give it a good shake, make sure that that bleach is in there. And now in order to basically do the, get it from that 1% or 0.1% to the 0.01%, I'm going to take a sample from the 0.1%, take 1 milliliter, and then transfer it over to the next tube. Put that pipette away. Recover the first tube, take the second tube, and then go ahead and shake that up. Make sure that's nice and shaken up. Uncover that second one, take another milliliter from the second tube, and then input it into that third tube. Once again, shake up that third tube. And then once again, take another milliliter from that third tube and input it into our fourth and final tube. All right, so once I have that fourth tube, I'm just going to go ahead and shake that up as well. So now we have our dilutions. We have the 0.1%, the 0.01, the 0.001, the 0.0001, and then we have our water, which is basically our control. So what we're going to do is we're going to add 0.1 milliliters, which is the equivalent of 3 drops. So 3 drops of E. coli into each one of these tubes. So I'm going to take a different pipette, kind of mix up that E. coli, make sure I get a good sample. And what I'm going to do is I'm going to take off of that first tube, just put 3 drops of the E. coli into that tube. I'm then going to do the same thing with the next tube. Another 3 drops into there as well. Same thing with the next tube. And then for the last and final tube, same thing. This one only has water in it. Once you have all of your tubes set up in that way, basically what we did is we added the E. coli to the bleach solution. So what we want to do is shake that up pretty nice, and then we're going to sit there and leave it for 30 minutes. After the 30 minutes, what we're going to do is we're actually going to take a sample from each one of the tubes and swab it onto one of these agar plates to see if any bacteria are going to grow. So you'll notice that I actually labeled my plates. This is the 0.1% bleach. So after 30 minutes, I'm going to take a swab and I'm going to swab a part of that onto that streak plate or onto that agar plate. And I'm going to do the same thing for each one of the tubes after that 30 minutes. So I'll have another video to show that process. But the whole point is right now we're allowing the bacteria to grow, that E. coli, in that bleach solution, and we're going to see whether the bleach solution is going to inhibit some of that growth, if it's going to kill some of the bacteria, and we'll see that by actually putting it on the agar plates and then putting those in the incubator for 24 hours and then seeing the results. If there was growth, that the bacteria survived and the bleach didn't kill it, or if there was no growth, that the bleach solution was pretty effective. So once we're done with all of that, we'll be counting colonies to get the results for our bleach lab.